So firstly, I just want to thank the organizers for inviting me here. It's a really great pleasure to be here, and I've got to say this is an extremely well-organized conference. I went to one a couple of years ago where they seemed to spend the entire budget on expensive coffee machines and the hipsters that you need to run them, and they forgot to do things like checking that the rooms weren't double booked and that kind of thing. So it's great to see a conference that's running really smoothly. So what I want to talk about is forecasting of ecosystems at a large scale. So it's pretty clear why we need to do this. So I think the diversity of life is one of the most striking features of our planet, and indeed perhaps the most defining feature of our planet, and it really defines us as human beings in terms of our relationship with the natural world, and that's something that I'll return to at the end. But that relationship with the natural world is clearly changing. Oh, I think this is the old version of the talk, so this is going to be interesting. Do you have another one here? Not a big deal. I didn't have any jokes prepared, though, so you're just going to have to wait, I'm afraid. Give it a shot. If it doesn't work, it's fine. That's fine. I'll just roll with it. We'll just go. Yeah, no sweat. Technical issues. We'll skip that slide. This is going to make it easy. So our relationship with the natural world is changing, and now human beings are really the dominant force in terms of structuring the surface of our planet. And last year, I worked with a lot of colleagues on assessing some of the pressures on the natural world. And I was just thinking about this yesterday and trying to figure out how to convert some of these units and values to something meaningful. So this is total global effort in bottom trawl fishing. And we were making predictions out till 2020. And the prediction is in kilowatt sea days. So the unit I converted that to is if you're pootling around in your little inflatable boat, to do an equivalent amount of effort as the global fishing fleet does in 1 year, you'd have to spend 3 million years at sea. And that's just the effort on the fishing grounds. That doesn't count the effort going back and forth to those fishing grounds. And humans are appropriating 17 petagrams or 17 trillion kilograms of net primary productivity from the environment for their own use every year. So again, converting that into units that perhaps we're more familiar with, that's equivalent to about 80 million blue whales and about 300 million elephants just left over. So we're extracting a huge amount from the natural world. So we obviously need to better understand and ideally forecast the impacts of our actions into the future. And there's an open question to my mind, is can we do this? Ecosystems are inherently complex and stochastic and noisy systems. So there have been a couple of papers recently that said, no, we can't. And I have some sympathy with this viewpoint. It's certainly a challenge, but it seems a little bit pessimistic to write off any possibility of doing so, especially with the gravity of the threats that we're facing. So I think we have to try, even if we don't succeed, and I don't think we should be afraid of failing if we can't predict at large scales. And one thought, if you can read that, one thought that I've had is, well, maybe ecologists get overwhelmed with complexity when physicists don't, because we can see butterflies and we can see the diversity and the sheer range of species and these unusual interactions around us, but you can't see atoms unassisted with the naked eye. So maybe we get overwhelmed by complexity. So to address this, we developed this concept called the general ecosystem model. So this is a large-scale abstract model that takes its lead from the general circulation models used in the climate modeling sphere. So most ecological models, and a good example of this is a species distribution model where you're trying to predict the range of a species. Most models are correlative, so you do a statistical relationship between, say, environmental variables and your species occurrences, and then predict its full distribution. So you're modeling patterns. If the conditions change, you have to extrapolate beyond the range of the data, and you're making various assumptions about how constant the processes are. On the other hand, process-based models, and a good example of this would be something like a climate model, is you're actually modeling the underlying mechanisms, the formation of clouds or the hydrological cycle. And this really gives you a better insight into the mechanisms and the interactions, and you're developing your understanding of the system at the same time as you're improving your models. And as I say, the GCMs, which started very, very simply in very, very crude constructs, are now extremely powerful tools that are obviously used by the IPCC in terms of projecting what our future climate will look like. There's a big challenge in that we don't have the equivalent of the Navier-Stokes fluid dynamics equations in ecology. So how do we actually go about doing something like this? Can we do it? Can we model the biosphere via fundamental ecological processes? Can we use a consistent model across land and sea? And can we make a model with predictive power? So can we use this to forecast changes even in a crude way? I.e., can we create a general or global ecosystem model? And again, I think initial models will be crude. There's no getting past that. Huge oversimplifications, but really what we're trying to do here is just start something and build a community. So what we did is we spent a few years working on something that we call the Mattingly model. And how it works is that it's a global model. It's spatially explicit. So we divide the world up into grid cells, a grid of cells, land and sea. And we have a philosophy. So we're trying to include all species, pretty much. Have a balanced consideration of trophic levels. So we're not just focusing on top predators or autotrophs. We're trying to include the whole range in there. And we're trying to be as open about this as possible. So the code is freely available. You can download it. You can try and play with it. I'm not sure how easy that is to do, but hopefully it will get easier. And you can tinker with it and change it. So it's reproducible. And a key concept of this model is emergence. So we want things to emerge out of the model rather than us hard coding them in. So we don't want to fix too many behaviors in the model. We want behaviors to evolve as a response to what we're doing. So how is this actually done? Well, fortunately, we have over a century, really, of quantitative ecological thought, metabolic theory, niche models, Holling's work. And then what we can do is we can blend all that theory together. Really, we're just gathering what's been done and synthesizing it. And then we put that into our world-renowned supercomputers. And we developed what we call the Mattingly model. I like this advert because this is called portable. But I was looking up the weight. It's 55 pounds. So this guy had to go through special training to be able to lift that and try to smile at the same time. There are some big problems when trying to develop such a model. So a key issue is that as we made this estimate of 8.7 million species around their routes on Earth, and the vast majority are undescribed. So how do we go about including all species in a model if the majority of them haven't even been collected? What we do is we model coarse functional groups, not species. So we use a set of traits associated with each organism. So it's diet, herbivore, carnivore, omnivore. It's thermoregulatory mode. It's mobility. It's reproductive strategy, and so forth. So each set of organisms has a set of traits associated with it that affect the role it plays in the ecosystem. And clearly, we cannot model every single individual. That's just impossible. We have huge densities of phytoplankton in water, or even of birds in a small area. So we have to aggregate somehow. And what we do is we aggregate individuals into what we call cohorts. So these cohorts have the same set of traits. They can be multiple species. And then we follow them through time. So they'll have things like how old they are, how big they are, whether they're growing or shrinking, what they're eating, how many individuals there are. They have a characteristic mass at which they achieve maturity and can produce new cohorts. They have particular prey preferences and so forth. So we have a bunch of these cohorts sitting in this grid in the model. And right now we're running, typically on a global scale, we run with about 10 million cohorts in the model. So the dynamics of the model, so this is, I'm not going to spend too long on this because I could spend a whole talk just talking about the model, and I don't want to do that. So I'm just very quickly going to run through the fundamental processes in the model. So you can imagine this blue circle is a cohort, and each black dot is an individual within that cohort. So at every time step, each cohort metabolizes, so individuals use energy and lose body mass if they don't have sufficient energy. They're predated upon, so the number of individuals can decline. They can eat other individuals and grow. If they're reproductively mature and have ingested enough mass, they can produce a new cohort. They go through mortality. So this is mortality not due to predation. So this could be things like starvation, background mortality, and so forth. And they can disperse, so they can move around the ocean or on land. And what happens is this set of dynamics is run for every single cohort, and from that, the ecosystem structure emerges. So from that, all of those predation events and the herbivory and the metabolism and so forth, you get characteristic structures emerging in terms of trophic pyramids, in terms of biomass density distributions, abundance body mass distributions, and so forth. And each cell in the model has a characteristic set of, I guess this isn't really working. So each cell in the model has a characteristic distribution that evolves over time. And then these cells are linked through dispersal. So I'm just going to give you quite a quick example of what we do for predation. So for predation, we don't have fixed links, right? We don't have a fixed food web. We have a dynamic food web. So individuals will feed based on the relative body size of the predator and prey, and the amount of any particular prey that they eat is dependent on the density of that prey in the environment and how close that prey is to the optimal body size. So for example, if there's very few optimal prey around, but lots of suboptimal prey, they'll switch what they're eating and move to a different kind of prey type. And so this produces nice emergent food webs where the structure isn't fixed and you can have really dynamic and changing ecosystems. And then we run the model. We have I realize you probably can't see this, but we have a complete plant model on land where resources are allocated to roots, leaves, and so forth. In the oceans, we're just using remotely sensed primary productivity to drive the model. And we have temperature, ocean circulation, and so forth. So environmental characteristics that we use to force the model. And so things like metabolism are dependent on the ambient temperature for ectotherms, and for endotherms, it's a little bit more complicated. So we start the model, basically put the same biomass, the same stuff everywhere. And what you get emerging, excuse me, is a characteristic distribution of biomass. This is just a plot of heterotroph biomass. And this reflects quite well the productivity patterns. You can actually see the seasonal shifts in heterotroph biomass as your model progresses. We run it with a monthly time step right now. We also get outputs at the community level, so things like characteristic distributions of body sizes, and at the individual level. We capture some properties quite well, so things like growth rates, which emerge as a balance between the amount of prey in the environment, the competition you have with other organisms, less your metabolic costs. So the empirical data in black, that which we could find in the literature, and our model outputs are in gray. So we're actually matching that quite well. Of course, some characteristics we don't match that well. That's just the nature of the beast, but then that tells us that something's missing or incorrect in the model that we have to look into. So that's a very, very brief overview of the Mattingly model. And so if you have this system, what can you do with it? What can general ecosystem models actually do? What are they useful for? Well, firstly, I think we've got to think about what separates them from existing models. So we're talking about something that's global in scale, land and ocean. And really, our focus is on ecology and biodiversity. We're not just focusing, on, for example, on several species of commercial interest. We're really interested in the ecological interactions and stability. And they're fully dynamic, so ecosystems can collapse. You can have trophic cascades. You can have complete turnover, and all of this it can emerge from the model. We try and include all species. There are some constraints on that, but so we're working with these functional groups to try and put everything in there. And you can have ontogenetic changes, so individuals can change their, for example, their dispersal patterns. They can go from being a planktonic organism to an adult organism which can advect itself, can essentially move itself around the marine environment. And we're also getting emergent behavior at multiple scales. So for example, you can see what lifetime reproductive success of an individual looks like, what your population dynamics look like, how do your food web and trophic structures look, how fast does energy move and cycle through the food web. And we can even go so far as to produce latitudinal gradients, which I'll show you an example of that later. And we can track all of these interactions, each of these events, and so we can do interesting things to do with sampling interactions between individuals. So this actually is something that we've been doing recently. There was an interesting paper in 2002 by Jennifer Dunn in PNAS, where she looked at essentially properties of food webs. And I'm sorry this figure's not very clear, but it actually isn't in the original paper. But what this shows is characteristic distributions of network properties in different food webs, ranging from grasslands to lakes to estuaries and so forth. And what we can do in Mattingly is we can sample individuals probabilistically, so you can take the chance of observing an interaction, the chance of sampling a gut. You can look at whether an organism has eaten another particular group, and we can actually produce exactly the same output, this whole set of distributions, just by increasing our sampling intensity, which I think is quite an interesting result. So clearly there's and this has been pointed out in the literature, but it's a nice demonstration that you really have to sample food webs quite heavily to really be sure about these network properties. We looked at perturbations and ecological collapse, so how far can you push a system before it collapses? We've primarily looked at this on land, and this is in terms of removing net primary productivity, so harvesting essentially. And what we do is we look at a particular place, and then we look at a range of functional metrics. So things like the body mass range, how diverse are your organisms there? What's their mean trophic level? Are you moving towards a system which is dominated by small-bodied, low trophic level organisms? And then we can see how that changes over on the x-axis you have your gradient of productivity removed from the system. And then we can repeat that for different environments with different climatic conditions. And so in some cases, you can see, so we ran a cell in the Libyan desert, and after about 30 or 40% of MPP removal, the system just collapses because there simply isn't enough to support the organisms. Another thing we've been looking at is hysteresis. So if you push an ecosystem with a pressure, does it return back to the same state? And if it does it follow the same trajectory back? Or do you need to invest more energy into returning the system to its original state? So we impact basically over a period of 100 years, remove up to 95% of the productivity, and then relax that pressure. We let the system go back. And generally what we find is that they don't return along the same. Firstly, there's a wide range of variability. So in some cases, they do return. So in B in France, you can see red is going out. So you're pressuring the system, and you're relaxing that pressure, and it's returning. And we can look at a number of metrics, but here we're just looking at biomass density. So here you can see it's returning to the same system, but following a different route. Whereas in other cases, in some places, you end up with higher levels of biomass. Again, you're transitioning to a system with lots of small individuals. or you can come back to a completely different system. So it's interesting for exploring those questions about ecosystem complexity and stability, which I think are really important for human purposes. That's bad news. So yeah, one crazy thing, okay, this is amazing to me, is we just literally, just for fun, we plotted the latitudinal ratio of endotherms to ectotherms in the model. So these are, on the left, we have the abundance ratio. This is in the oceans. The abundance ratio, so on the right is the abundance ratio. So clearly the system is dominated by ectotherms. And then we have the biomass ratio, so still dominated by ectotherms, but a much narrower gap. And I was thinking about this and thinking, well, this is really interesting. Let's find out if it's right. And so I looked through the whole literature. There's nothing on this. There's nothing on this really, really basic kind of detail about life on our planet, about how the relative ratio of ectotherms to endotherms, either on land or in the oceans. So I thought that's pretty crazy. So we can generate these hypotheses, and maybe they can go out and people can test them or provide more information so that we can start to check and see if what we're seeing here is reasonable. But ultimately, what we'd like to do is we'd like to be of relevance to policy and to decisions that have to be made about conservation. So what we're doing is we're rolling back the model to 1859 using Earth system model climatic outputs, and then running forward to the present day. So we'll see how ecosystem structure has changed in the last 150 years or so. What would ecosystems have looked like 150 years ago, and what will they look like almost 100 years in the future? And see the baseline, the present day, and the future of ecosystems. And this is ambitious. This is a way off yet, and I'm kind of very slow at getting around to this, but it is something that we're trying to work on. And ultimately, we'd like to look at the big picture. So what are the trade-offs between agriculture and fisheries? If you increase your agricultural intensification, can you decrease your level of fisheries? And if you can do that, where is the best place to do it? How do you spatially make those trade-offs? All right, just conscious of the time here. So DNA barcoding, I think, is from what I've seen, and I've been really interested to attend the talks here, and they've been phenomenal, and I've learned an awful lot. I think DNA barcoding could be a really valuable tool to feed into ecosystem models, because one of the things that we're finding, and I can skip that slide, is that it's very, very difficult to get data against which to compare these models. Because what typically happens is that what has happened is that ecologists go out, they have their target species, they sample an area, typically less than 5 meters squared, and they keep doing this. And even if they sample a larger area, they may just look, for example, at all the autotrophs. They may be interested in the plants, or they may be interested in insect-plant interactions, but not look at birds or mammals. So what we really need is a synthesis of information from the top to bottom of ecosystems to really enable us to constrain these models and really start to build some kind of understanding about their capability for forecasting. So I had a look in the literature. On the other version of my talk, there's another slide here, so I'm just going to discuss the slide that you can't see. It's about Eniwetok Atoll. So in 1955, Odum and Odum went to Eniwetok Atoll and sampled the reef intensively, top to bottom, before a nuclear weapon was detonated there. So that was a place where there were a lot of nuclear weapon tests. So that's one paper we found where there's relatively good top to bottom information. Another example is the International Biological Program, where there's actually pretty good sampling of entire systems of the African savanna. Okay, so looking across different species groups. But really, there hasn't been much else apart from those two examples that I could find. In the oceans, we have trawl surveys, which are pretty good, to be honest, but they're not close enough. They may just focus on species of commercial interest. They might not be interested in marine mammals or zooplankton or some species of crustaceans that have no relevance for them. So we really need to, from our perspective, it would be really great to do these kind of top to bottom sampling of ecosystems. And what I'm wondering, and really the question I'd like to throw out here, is whether meta-barcoding and eDNA can help with doing that, because everything I've seen suggests that it could be an amazingly useful tool to generate this complete ecosystem structure. So here's my wish list, and this is a wish list, okay? It's not gonna happen. I want the biomass of every functional group and every trophic level, relative abundance, the function, that can be very coarse, so it can be things like endotherm, ectotherm. It doesn't need to be very specific function. Material flow rates, top to bottom of an ecosystem, repeatedly, and ideally I'd like it by next weekend. Oh, I've been shot down. Oh no. I'm almost done. There we go. Yeah, okay. So I would love it if, when thinking about the next 10 years of the barcode of life, you really thought about whether any of that information can be collected and included on the pages with each bin. They're really trying to kick me out of here. I'm almost done. Please, just like 2 more minutes. I need a coffee as well, so I understand your impatience, don't worry. Okay, we're on down here. So I'm just going to talk you through the last few slides. So in the shorter term, can we think about looking at DNA sequences to give us some function? So I know this is done in the microbial world, but can you derive function from some of the DNA sequences, even at a coarse level? And one thing that I think would really be helpful for us is to categorize organisms at a higher level. So this was mentioned actually in the last talk. If we can get information quite easily about a family or an order, actually the species identification is it's really interesting, but it's not necessarily that important in terms of the function of an organism. So that's fine. And in the future, the second last slide, so for example, I'm getting involved in a lot of projects where we're attaching acoustic tags to sharks. So you attach a tag and it communicates and tells you where the shark is moving. So what happens maybe in 5 or 10 years when you have these portable samplers? Can you have a sampler attached to an acoustic tag that just filters the water as it goes through and looks for different environmental DNA signatures, whether it's from a particular species of interest or more broad spectrum? I think that's really, we should start to think about the long term. And the last thing I want to say is I was really tremendously excited by Paul's description of a planetary biodiversity mission. I think that's a really wonderful and ambitious project, and it's the kind of thing that we need. But I would emphasize that from my perspective, there are all these add-ons that you can do for just a little more effort. So you can, as well as cataloging species, you can feed into modeling efforts or policy efforts and really do that and really get a lot more out of it for just a little more effort. So I think we have to do this for humanity, both for humans as a whole and for our humanity, right? We have to look after the natural world. And for life, because it's for biological life. Such a mission is for biological life, but it's also for our life, and we're really dependent on that. And that is it. There we go.
Thank you very much, Derek. There is time only for one question.
Coffee. You can ask me at coffee. It's fine. Yeah.
Okay. Thank you very much. We are coming back at 11:15. Thank you, Derek.
It's fine.
apologize for that.
Don't worry
I have
about
no
it.
idea.